So today we are going to discuss about the drugs and pharmaceuticals. So what is mean by drug? So generally you can define the drug where the drug which cures a disease. Simply you can define the drug where the drug is okay. So which cures a disease. Okay. So drug means which cures a disease here. So what is mean by disease? Okay. So what is mean by disease generally? So generally the disease nothing but where the alteration of biochemistry is a disease here. So where alteration means the changes. You can see. Okay. So the changes. Some changes in the biological system is called the disease here. So in that case, means the changes in the sense the deviation, right? So the deviation in the normal biochemistry, right? Alteration of biochemistry is disease here. So when we get the alteration in the biochemistry, we use drug compounds to bring the deviated system into normal mode. So that is called the drug here. And the drug, we know that so the drugs are various types here. Okay, so here you know that the drug used today in medicine are either obtained from the natural sources or it's synthesized. Right? So we are using the drugs by two means where some drugs are synthesized artificially in the laboratory and some drugs are available are extracted from the natural sources here. Right? So generally we call these are the natural drugs which are obtained from the natural sources called the natural drugs here, which are obtained from plants, animals, and marine organisms here. And the drugs from microorganisms are the dates supportive addition known as antibiotics here. So because you know the history of antibiotics. Yeah? So where the history of medicinal plants is it is an old history of mankind here. So you know the how the antibiotic like the penicillin was invented here, right? So we are having so many types of drugs where used to cure various symptoms and various diseases here. So you can use drugs for vomitings, and you can use the drugs from diarrhea, and you can use from some fever and some other things here. Okay, so and in this particular drug chemistry, we have some terminology where which is involved in the drug chemistry. You see the drug, already I told you the drug nothing but which, okay, the drugs are defined preciously under some acts here. So, where which cures this disease is the drug component, and basically the drugs and the disease, it is both are interrelated here. And disease word is used to indicate a state of want of health in mind or body here. Okay. So in fact, this word is a combination of this and is. Okay. So here the disease. Okay, the disease name. This is a combination of the this and is here. So where it is these two, this and is. This is indicating the absence of uh, easiness or the well-being or a feeling contrary to the feelings of health common about due to abnormal function of body parts here. So it is simply called where the deviations, nothing but where the illness or unwellness is called the disease here, unwellness. So when you are not feeling good, when you are not feeling uh, comfort, then that condition is called the disease here. Okay, then the pharmacy. So you know that the medicine, okay, and the pharmacy, both are somewhat, both are interrelated only, but where you see this is a branch here. It is a broad field. The pharmacy is a broad field here. You see the field, it deals with the formulation and the standardization of drug called the pharmacy. Okay, so here the pharmacy, nothing but where it deals with the formulation and the standardization. Generally, with the drug compound which is synthesized in the industries or the laboratories here, and after synthesizing the drug, where undergoing some right, it undergoes some test here. We call that is a standardization. Okay, like you know that there are several stages of there in the drug development or the drug discovery, where you can see the identification of the right identification of target here, then choosing of right, so the disease, okay, and then the development of a active lead compound here, then the lead modifications, okay, then key clinical trials here, QSR studies here, then lead modification, and then we go for that marketing, means approval of the authority bodies. Like we have various approval of authorities like API, okay, IND and NDA. So all these are the various authorities which gives approval to your synthesized or the new drug compound here, right? So then we go for this particular statutory approvals, right? Then it will be undergoing the examination generally, and by approval of these authorities, then finally we go for the marketing here. Okay, so and you know that in the clinical trials also we have various stages. Like there are seven stages of clinical trials. Clinical trials 1 and clinical trials 2, 3, 4, 5, 6, 7. So there are seven stages of clinical trials. So preclinical trials, post-clinical. So all these are the stages of your drug discovery. So where pharmacy, it deals with the synthesis or the formulation of the drug compound here. Then the standardization of the drug. It is called a pharmacy. Okay. And the main aim of this pharmacy to serve the society with great professional responsibility in the appropriate use of medication here. Okay, so you know that because where we are synthesizing or where we are inventing the drugs, we to serve the nation or to serve the peoples who are affected with the various with chronic diseases here. Okay, so all these things here. Yes, and here the devices and services to achieve requirements of therapeutic treatment. Next, pharmacology. Pharmacy, it is a broad, it is a field of that which deals with the formulation and standardization. And the pharmacology, that word pharmacology is derived from the two Greek words like pharmakon. Okay, the pharmakon, which is means the inactive here, inactive, inactive principle, inactive principle here, and the logos means disposer. Okay, so pharmacology covers the physical, chemical, and biological and physiological effects of your drugs here, right? So where it pharmacology where it deals with, right, it deals with the where the activity of your drug compound here, right, the where the activity, like it is physical activity, chemical activity, and the biological activity. So all these activities is deal by your pharmacology. It is one of the subject or the broad field here, where pharmacy deals with both the formulation standardization, yeah, and your pharmacology, it deals with your where the all the properties of your drugs. Okay, like you see that ADME properties. Okay, so you know the ADME property where absorption. Okay, so ADME property. You know that ADME absorption here, distribution and metabolism, excretion, toxicity. So these are the five properties of a drug compound here. So you see your pharmacology is dealing with your uh, all these properties like physical properties, chemical properties, and biological properties here, which affects of your drug compound. And it explains the mechanism of action also. Means how what is happening in what will happens when you consume or when you take the drug compound here. So that thing, that entire mechanism is deal by your pharmacology. Then the pharmacophy. So then you know the pharmacopoeia, it is assembly. Okay, so it is uh, enable of steric and electronic features that is necessary to ensure the optimal supramolecular interaction with a specific biological target and to trigger its biological response here. Next, pharmacodynamics, very important terminology here. Pharmacodynamics and pharmacokinetics here. These are two important terminologies, which you see the pharmacodynamics, where it deals with the right pharmacodynamics, which deals with your uh, with the journey of your drug here. Okay. Next, so here the pharmacodynamic, it is characterized the relationship of your drug concentration at the site of action to intensity of biological response here. Next, pharmaco phase, pharmacodynamic and pharmacokinetic. These are the important terminologies, two phases of your drug. So where both will deals with various parts of your drug here, where you it is right the pharmacodynamics, which deals with the journey of drug, and the pharmacokinetics, which deals with the action of your drug, right? The response and action. So when we are designing the drug compound, where we will decide the dose and dosage form here. Okay, so dose. Generally, you know that the dose when the drug is invented. So after means after approval or the authority, right, the statutory bodies here, then it is going to decide the dosage decides. So generally, the dose decides is the dosage form is decided by according to your FIFA rule. You know the FIFA rule. So FIFA, as per this FIFA rule. Okay. So as per this FIFA rule, we can decide the dose because you know that the drug compound. It is different for right. It is different for race and different for gen. What is that? Sex and different for adults. Means it is different for adults and kids, and it is different for women's and different for gents. Okay, so all these are the various right the criterias, right? Various criterias uh, which is decided your dose from. Yeah. So where your pharmacokinetic studies which deals about all these criterias here, right? And you say that pharmacokinetic studies look into these aspects like how the drug is absorbed and metabolized and distributed and eliminated in the body. You know that we call this as the ADME studies here, right? So where this particular pharmacokinetic studies, it deals with the ADME properties. And you know the ADME properties where the A is absorption here, and D is a distribution, and M is a metabolism, and E is a excretion here. So these are the four terminologies, four properties of your drug compound. So absorption means when you take a drug compound. So you can take the drug by orally or intravenously, where we have two administration methods where to take the drug compound. So you can take the drug by oral form, means you can take from the mouth here, and you can administer the drug by intravenous, by injection form also you can take the particular drug. Right. So whatever the administration method is that, but when you are taking the drug, right? So it will be absorbed through your body tissues. Okay. So it will be absorbed through your body tissues here. Then after absorption, because you see where the body tissues are, it is hydrophobic nature here. Okay, hydrophobic nature. And here the distribution. So after absorption of your drug compound, it will be distributed throughout your body. Okay. Right? So after absorption, it will be distributed throughout your body here. Generally, it distributed and it is reaches to the site of the action where the diseases occurred. So it will be reaches to that particular center or the site where the diseases occurred. That is a distribution. Okay. Then the metabolism. So metabolism, nothing but where you when you take a drug compound here, so it will shows its effect to a particular site where the diseases occurred here. Then the drug compound, due to some enzymatic reactions or some acids, bases present in the body, so it will be react with that, and it is going to convert into another form here. Generally, you know, where the metabolite, it is not having any pharmacological other drug properties here. Non-drug, we call it is a where the drug compound is converting into non-drug compound here. That is a metabolism. Then excretion. So where the excretion, nothing but when the drug compound is converting it to metabolite here, and when it is showing its effect here to site of action, then it is going to excreted or the eliminated or the removal, right? So where it will be excreted through your renal system or the urinary system here. Okay, so these are the four properties. And when that a new drug or any drug compound which is invented or introduced here, so it must be having good all good all these admit good properties here. Okay, it must contains a good absorption property, good distribution property, good metabolic properties, and good excretion property. Okay, so then only you the drug compound said to be the ideal drug or the good drug here. Okay. Yes. Okay. Then the pharmacodynamic phase, where the concentration of drug at the site of action is monitored, as these are related to the magnitude of the effect produced here, the time course of intensity of this therapeutic and the adverse effects can be evaluated. And you can see a schematic diagram of your the drug action here, how these particular ADME properties are employed. So you can see whether dose of drug administered here. So dose of drug administered. So you can right. You can take the drug compound. The administration is by two means, like it may be orally or the intravenously here. Then it will be absorbed here. The absorption. So it will be absorption. It is absorption, not adsorption here. Okay. So absorption, where you know that the difference of absorption and adsorption. Okay, so where the adsorption is a internal adsorption is a surface phenomena, and absorption is a internal phenomena. So where your drug compound will be adsorbs in the body tissues here, then the drug concentration in the systemic circulation, right? And it is reaching to your the tissues, means the target. We call this the distribution. Okay, and that particular, right? So distribution. So you see this. It is the equilibrium stage is established here, and then the drug concentration at the site of the action. Okay. Then you see the pharmacological effect here, and then it is showing your clinical response. Okay, so the clinical response, and generally you see when you are taking the drug compound, so it may have the effectiveness, or sometimes some drugs will shows the side effects. We call this is the toxicity. Okay, we call this the toxicity, where that it is showing the side effects here. Okay, so like you see any some so many symptoms you can see, that is some allergic conditions, right? So unconsciousness. So all these are the some side effects which is caused due to that the adverse effect of your drug compound. And you see this the above one, all this called this pharmacokinetics, right? And the below one is called this pharmacodynamics here. Right. So then you see this receptors, the terminology where the receptor. So the site where the drug molecule is recognized and gives desired activity called the receptors here. Right. So drug receptor, we know that drug receptor interaction. So when the drug compound, generally the drug action, right? The drug action or the mechanism is depends on the drug receptor interaction here. Okay, drug receptor interaction. So when your drug compound is interacting your receptor, here the receptor nothing but where it may be the protein part or the protein molecule or the enzymes, are so many types are there. Enzymes, proteins, or any other like carbohydrates or the ion channels. So these are the receptors here. So when this particular drug compound is right, the drug compound is interacting with the receptor, where this receptor is recognized and it gives the desired effect or the activity, where it is called your receptor, means which recognize the drug is a receptor, nothing. And the metabolites, where the metabolites are the intermediate products, it may be the endogenous chemical or the exogenous chemicals. Okay, the drug compound. So you see this endogenous chemical, nothing but the neurotransmitter. Endogenous chemical, nothing but your the neurotransmitter, where which is already present in the body, and the exogenous chemical where you are taking from outside. So it may be your drug compound. Okay, obviously it is big drug compound only where we are using from outside here. Okay, so and the metabolite regulated by the various enzymes, so that naturally occur within cells here. And you see this metabolites are the two types, where the metabolites produced in the living cells and are useful to the cell functions, and the metabolites produced from the drug when they are acted upon the enzymes. Okay. So here the metabolites are two types generally, where already which is present in the living cells and which are useful for the cell in functions, and sometimes where the drug compound, right, the metabolite produced by the drug compound. Next, antimetabolites, where this antimetabolites are the drugs that mimic the natural substance metabolites of an enzyme. Okay, it is mimicking the action of your drug. Okay, it is not the actual uh, metabolite here, but it is mimicking. Okay, so the metabolites are drugs that mimic the natural substrate of an enzyme, that enzyme involved in a specific biological or metabolic pathway. It is inhibited. Okay, that is metabolite like drug molecule is incorporated into endogenous components. Then the knowledge of medicinal plants. Okay, so in the ancient, in the olden days, okay, so there is no development of the science. There is no development of the technology here, science and technology. So in that time where there is no the means medical field or any the medicines are developed here. So only that plants are described in Rig Veda and Ayurveda. Okay, in you that is the period of 3,500 to 1,500 BC, yeah? Which is we are mentioned in the Ayurveda. You know that Ayurvedic is developed where it is written by your Charak Samhita, and right, so written as the Charak Samhita, where it is dealing mostly with the plants and some plant medicinal importance here, plants which are having the medicinal importance, which is deals with the surgery also, the well-known ancient okay, thesis in the Ayurveda. Okay. So the bark of Saraka indica, you know that Ashoka trees, which are uterine tonic, and it is dried roots and stems of Vithania somniferum, Aswagandha, as the sedative. Okay, so in the Charak Samhita, it is mentioned about the medicinal importance and the properties of your some of the uh, plants which is extracted from that particular plants. You see that Ashoka, Saraka indica, Ashoka, where it is having the importance and having the pharmacological activity of your uterine tonic here, and the dried roots are the stems of Vithania somniferum, which is called the Aswagandha, right? So it's the roots, the active compound which is extracted from this Vithania somnifera, which is acting as a sedative, and it is very ancient. It is known to the ancient Indians here. Next, development of materia medica and pharmacopoeia. Here, the Hippocrates in 460 to 370 BC, The Greek scientist was considered as the father of medicine in the Western world. He's a Hippocrates. Hippocrates is a father of medicine, uh, and he recommended the use of metallic salts here. Aristotle, the Aristotle is one of the famous uh, scientist here. He's a student of Plato and philosopher. Right, Aristotle is one of the great philosopher here. He's a student of the Plato. Okay, Plato also one of the philosophers. So Aristotle is a student of this Plato here, and his writings are known as Animal Kingdom and Theophrastus. Okay, a student of Aristotle wrote about plant kingdom. You know that plant kingdom we have like arthropoda, mollusca, annelida, okay, amoeba. So all these various plant kingdoms are there, right? So mollusca, nematohelminthes, okay. So prokaryotes, eukaryotes, all these are mentioned in his writings here. He described and wrote about plant kingdom here. Okay, that Theophrastus, who is a student of Aristotle, he was mentioned and he was written about the plant kingdom. Right? And that another, right? So that Roman naturalist Galenius, he also devised methods of preparation of plants and animal drugs known as Galenicals. Okay, so all these are developed the materia medica here, and the necessity to maintain the purity of drug and in his intimation of the dosage levels finding is beginning with this work here. So many new drugs of botanical and mineral mineral origin came into this use. So several you were using various medicines or the drugs compounds which are in the pure form, where means all these are the mentioned and invented by all these philosophers and the scientists here. Next we'll go for this chemotherapy and synthetic drugs. Okay, so in the olden days, where in the BC, there is no development of the science and technology, so they used to use the they are curing the diseases by chewing the uh, leaves of the plants and okay, so and the chewing the roots, leaves, all these. Right. So the history of chemotherapy falls into three phases where okay, chemotherapy nothing but where the treatment of this disease by the chemicals here by using that synthetic drugs. So here the history of chemotherapy. So it is divided three phases here, where that early period of empirism is upon the discovery of salvin by Eldridge Hatta in 1905. Right. So then in the period of up to 1935, so is Dombik introduced the proton seal, and then the third phase where the modern period of that sulfanilamide as antibodies. You know that sulfanilamide is one of the great antibiotics here, which simultaneously which is isolated of few drugs from long known natural sources and which is synthesized by the laboratory techniques where it is used in this synthesized in the 19th century. And by development of the sulfonamides, various drugs are synthesized like acetonilide and salicylic acid, which is synthesized by coal-based synthesis in 1818 to 1894. And the antipyrin with the Knott scientist was synthesized in 1883. Then very antipyretic drug like aspirin, which is synthesized in the 1899, and barbital, Emily Fisher, right? Emily Fisher and Mering in the 19th century was synthesized. And the first hormone with adrenaline and that local anesthesia. So all like benzocaine. So all these drugs are synthesized in the progress of these sulfonamides. Yeah. Right? In continuation to this sulfonamide, it is antibiotics here. Various drugs are synthesized. Means antibacterial, antifungal, and anti anesthetics, and okay, adrenaline, the hormones also synthesized. So all this the development in this drug chemistry is developed, means medicinal chemistry is it is takes place here. Then the Louis Pasteur, that disease was caused by the pathogen parasites due to cause of chemotherapy of healing with chemicals. You know that Louis Pasteur, who is a microbiologist, we invented the penicillin. And you know the penicillin invention is done by accidental. We call this uh, serendipity of drugs here, right? Where what is mean by serendipity of drug? Where unexpectedly invention of drug here. Unexpectedly invention of drug is a serendipity of drug. So we have this generally, you know, the Louis Pasteur. He is a microbiologist here. He was done. He means his aim to develop a bacterial culture, and he kept some bacterial culture in the petri dish, and he forgot about that particular experiment, and he left that experiment here. Okay, after few days, like it may be weeks, right, a few weeks here, he forgot about that, and he was busy with another work. Okay, so he was busy with another work, and he forgot about the earlier experiment, which is kept to develop the bacterial culture. And on one fine day, he was accidentally he was seen that the earlier experiment, and he was observed. Okay, when he observed that petri dish, yeah, right. So the bacterial development is done, and he found some fungus formation is done here, right? So he found some fungus formation, and he observed that where that the few of area is in few of area in some areas there is no development of bacteria here, and in some areas the bacterial development is done. So then he was thought that something is there, means some active means some pharmacological activity, some active compound is present in that particular what is that you call that fungus, right? Where the fungus is having some active means activity here. Okay, that's why the it is preventing to develop the bacteria, because where the fungus is developed here, there is no development of the bacteria. So where he thought that there is a some activity is there which is preventing the growth of the bacteria, right? So later it was discovered that where that active compound which is produced by that fungus is a penicillin. Okay, so it was an active compound developed by means it is active compound is found as penicillin here, which is antibacterial drug compound. Okay, so in this way it is okay, where they are discovered the penicillin here, and it is okay. Then that the research activity shifted to a cooperative effects of groups of scientists and industries, laboratories, rather than the individual. Okay, so in this way, the development of the drugs, medicinal chemistry is done.